Okay, great. So I'm going to open by telling you all a little secret. So when we first envisioned office hours session, we were thinking it would be kind of an intimate group. This is sort of an early feature. And, just a few folks would get together and I would be able to show you what we have that's really in production. It's in development. This is a early release. We want to get it out. So it's in people's hands. It's helping you answer questions, but with the understanding that, we're still refining it and getting it to a good place. Well, it's not quite as intimate as I had imagined, but the product is still in that space. So come to this with the early access kind of attitude. This is a thing that we want you to have value for you immediately. And we also know that there's a long way to go with it. So I want you all to be a part of helping us figure out where we want to go. All right, so let's actually start looking at let's start looking at our product. I'm going to share my screen, and that's another technical challenge. We'll see if it works. All right. So here we are. This audience I know is kind of a mix. We have some old Plasmidsaurus friends. Not that you're old. You've been friends a long time. And I'm sure we have some newcomers too. And so I've selected topics, and I'm kind of trying to use a pace that's going to meet everyone's needs. If you're very familiar with this product, I'm definitely going to tell you some stuff you already know. And if you're brand new, I might gloss over some things that are still important. So by all means, use the question box, and we'll hope that everyone can learn everything they want. So here we start at the Plasmidsaurus website, and we have a number of different products, not just plasmids. And what we're going to talk about today is in this category of whole genome sequencing, and we're going to talk specifically about bacteria. So let's look at what that product is all about. So first of all, it's got happy dinosaurs, because all Plasmidsaurus products need that. So bacterial genome sequencing is what you would expect. You send us bacteria and bacterial DNA, and we will send you back an assembled sequence. And like all of our other products, the turnaround time is very fast. In many cases, it can be a day. If it's a very big run, it could take a little bit longer. And if you want us to extract the DNA, that can add a little bit of time too. So the nice thing is there are many options for many use cases. And as I suggested, you can isolate the DNA yourself and send that to us, or you can grow your cells, spin it down to a pellet, put it in the Zymo Shield, and we'll do the extraction for you. So whichever version is most convenient for you. As you're thinking about ordering, just some features of the product, there's a lot of information on this page. What to expect, the date that you get back. This is a lot of what we'll talk about today. Many more details about how to prepare your samples and how to send them to us, either for the genomic DNA or if you're sending a bacterial pellet. It's really worth spending a little time on this page and getting the information you need. And then lots of FAQs at the end, lots of questions. So you've prepared some samples, sent them to us, and then what happens? You get an email back that says your results are ready. So when you see this, it will remind you what you ordered, whatever you happen to have called that order, and give you some options. There'll be a button right here to view your results. That's the page we're going to spend most of our time on today. There's also a button to download directly. So let's go to that order results page right here. And here's what we've got. So if you have ordered some bacterial genomes from us in the past, you may see some new things. Let's go over it all from top to bottom. So what do we have? We have the name of the order. This you entered when you placed the order with us to remind you which samples these are. Up at the top, we've also got the same download results button that we had in the email. So these will be all of your analyzed files. These are the sequences, a number of plots, some tables of summary data, everything that you could want from the bioinformatics pipeline. And the separate button for the FASTQ files, you can also get all the raw reads. But what can we do from the website before we click anything and go anywhere else? So we have some information about the whole order at the top, just a unique identifier, the date, we have the product that was ordered, and the number of samples. So one of the fun new features that we have for bacterial genome sequencing is this order summary figure at the top. So we had 4 samples. These are the 4 sample names. And what we've got here is a tree and a heat map of similarity to each other. So this gives you a quick glance, some initial information about how similar your samples are to each other. In some cases, you're sending a batch and they all are meaningful to each other. In other cases, if it's a sort of grab bag assortment of what needed to be sequenced that day, this may be less meaningful. But a quick look anyway. So just a review or a little bit about how to interpret this heat map. The color indicates how similar 2 sequences are. So the light here is 1, they're identical, and the very dark at the bottom is they're completely different. And so each row, guess, corresponds to each of these samples. And then as you think about the columns, if you kind of mentally rotate that set of sample names, you'll get the pairwise comparisons. And the way I remember it is this diagonal here is 1. So that's the cell that compares each sample to itself. And of course, it's identical. So this is the comparison of this first sample, NEB Turbo, to itself. And then this is the comparison to the one just adjacent to it, and so on down the list. So that new feature, happy to share with you. Hopefully it gives you some quick insights right from the website before you have to download anything. So the next section of this page is the information for each sample. And each box represents 1 sample. So we have a name that we gave it, just basically just numbered 1 through 4 in this case, the names that you gave the samples. So this one is in fact a sample from NEB, the 5 alpha strain. And then this little check mark means that the sequencing was successful. We got reads, we got an assembly, it looks good. So in the past, this has kind of been the extent of what you get. You have a list of your samples, it worked or it didn't, and you have this nice button that lets you dive into the results. So what we've added in the last couple of weeks is this additional summary information about each sample. So very quickly, you can see how many total reads did we get, what's the depth coverage, and what's the length of the total assembly. Equally important is this list here of how many contigs were created when the sequence was assembled. So here we have 1 contig of, here's the length. And then part of the analysis process and part of the results you get when you download the full package is this summary, this analysis too of what species we found. So in this case, it's an E. coli K12 strain, which is exactly what we'd expect as this strain that we've ordered from NEB. So again, quickly, you get the basic information about your sample without having to leave this page. And here's the showstoppers, what we're all waiting for. We also now have a feature where you can compare your assembled sequence to a reference. So let's go through that process now. The first thing we're going to need to do is upload a file that has our reference sequence. So we're going to scroll to the bottom of the page here, and you'll see this section, which is new for bacterial genomes, but might look familiar if you've ordered some of our plasmid products before. So for a while, we've had the ability to compare your plasmids to a reference sequence, and now we're extending that to bacterial genomes. So you'll click on the button, and I mean, everyone has uploaded a million files at this point in their life. You'll select the one you want and click open. We're not going to do it live now for 2 reasons. One, just anything live is terrifying. But also you may have noticed in that email, it said, "Hello, Matthew." So this is a sample, or excuse me, an order that's been shared with me. The feature that's available to anyone. If you place an order and you want your PI, you want your collaborators to have your results, you can share it with them and they can go directly from their account to see it and play with all these features too. However, the person who placed the order is the one who can upload these files. So Matt very kindly uploaded a bunch of files for us. And here's the first one we're going to use. It's called the NEB5A sequencing results January 25. So let's imagine we're a lab that likes to check on our stocks with some frequency and make sure that, we don't have contamination, we haven't mislabeled the tubes, etc. So we sequenced this strain 6 months ago. We're doing our regular check now. We're going to compare to the results that we had last time. So we just select that file that we want and we click visualize. And it's going to do its thing and show us a nice plot. Now, again, if you're familiar with the plasmid product, you'll notice that with that one, you upload the files and the comparisons happen kind of automatically. And here we don't want to suppose which comparison that you want to do. You get to say it explicitly and then click the button so it does the alignment. So here we have it, a nice visual. What we have at the top is a line that represents the assembled sequence. This is the name of the sample. And then at the bottom, we have a line that represents the reference sequence. This is the name of our file. And in between, we have these gray lines that tell us where the alignment, where the two sequences match. So as we might have hoped, this is a perfect match. There are no differences between these two sequences. Our sample is in good shape. We got the answer we need. We're done. Fantastic. And as we all know, having done lab work for more than 10 seconds, that's not always the case, right? Often things are more complicated. So let's look at some more samples to see what other kinds of visualizations we might get. So here's the second sample in our this is also an NEB cloning strain. We can look at our assembly. It came together nicely as one contig, identified as E. coli. And what we're comparing it to here, this is the reference that was published by NEB. So they submitted this sequence to NCBI, and we went to the NCBI website, downloaded the sequence as a GenBank file, uploaded it to the site here, and performed the alignment. And here we can see that there are a handful of variants. So 6 variants altogether, and they're represented by these lines. So an orange line means there's a SNP. There's a single base pair that differs between our assembled sequence and our reference. And the yellow lines tell us that there's an insertion. So there's an extra 1 or more bases in our sequence or an extra 1 or more bases in our reference. Of course, that means there's a deletion on the other one, right? So as we present them in the table, you'll see 3 categories. Deletion, basically meaning there's an insertion on the reference, a SNP, meaning an identical or a change in the same length, and then a insertion, meaning more bases on our sequence. And in the table down below, we get a text summary of what we can see in the picture. It's going to tell us which contig the variant is on. It's going to tell us the type, the start position and the end, and these coordinates are on the assembled the assembled chromosomal sequence. And then the size, so you don't have to mentally do a lot of integer arithmetic. And where we can, we'll also identify which genes are affected by this variant. Here we have a case where there were 2 different genes cut out, deleted. And here we have a case of a couple of insertions that happened between genes. So there's no annotation to report there. So in this case, a little bit more complicated story, basically aligns very well just a handful of small variants. So let's look at something slightly more complicated. Here's our third sample, also an NEB strain, the turbo strain. So this one ships with the F plasmid. So we know that our assembly should have both a chromosome, and then we know there'll be a bunch of reads that are not part of the chromosome. And in fact, what we get is one nice big assembly that matches E. coli as we would expect. And then we get these other 2 contigs. So the way that our particular pipeline works today is that it's very oriented around making sure you get a great assembly for your chromosome. It is not also tuned to make sure that all the plasmids assemble in total. But this is on our backlog. We've heard from folks that this is an important component of their sequencing result, and so we're going to update the pipeline for sure. In fact, I'm pretty sure the engineer just told me yesterday that the work on that got started this week. So that's exciting. But in the meantime, this alignment feature gives you a nice way to check that, yes, these two regions, these contigs that weren't part of the chromosome, are in fact part of the plasmid. So I chose a reference sequence that had two sequences in it. It has the plasmid and it had the chromosome, so they both show up in our figure. And then all three of our contigs also show up in our figure. And the gray lines just indicate more of where the alignments work, so which sequences match each other. And in this case, these two both match the one reference sequence. So it's a visual that gives you quite a bit of information at a high level, a lot more to infer about what these three contigs are and how similar they are from the published reference. Overall, very, very similar with just a handful of SNPs and indels. So let's get a little more complicated still. Here's our fourth sample. This is another cloning strain that we ordered, and again, it's the published reference sequence. And here we have a very big rearrangement. So we still see some yellow and orange lines, but there's this whole region of our assembled sequence that actually aligns to this other section of the reference. So the diagram information that we have lets us see that pretty easily, which is nice. This is mostly to say, if you do align your assembly to something that's quite different, you'll still see that in the figure. It's not like the figure will become completely uninterpretable or just not work. We still get the table of variants, and this one's quite big. We don't show all 213 on this page. That would be an insufferable amount to have to scroll, but instead you can, in fact, click through the table and look through all of the variants here. And in addition, you can download just this table. So you can open it in a spreadsheet, sort it, search it, look through it to see, are the genes that you're interested, the ones affected, and so on. So that's our fourth sample. I really wanted to focus today on the newest features that we have and the ones that give you a very quick look at your results. So before you want to do your in-depth, your deep dive on your data, here's quite a bit more information you can get just from the website. In many cases, there's still more work to do, and so this is when you would download the results or perhaps click on the View Report. There's a lot in this report. I'm not going to go through it all, but let's definitely just take a peek. So now we're looking at a whole page that's all about one sample, and we've got quite a few figures and tables here, and there are files in the download that correspond to all of these. So a Circos plot that gives you a very high-level view of all your annotations, some statistics about the assembly and how the sequencing went. This is called a bandage plot, which has to do with the structure of the assembly and the pieces that were put together. We've got more on the species analysis. So some of this was summarized for us in the table, and this is a little bit more information about it. And a very nice figure about the reads, what is their quality, what's their length, which ones were down selected. And then at the very bottom, we also have a lot of explanatory text, how to interpret these figures, what am I looking at. So there you go for the view results page as well. There's one of these for each sample. And that is just about to conclude what I had for the part that we're going to record. We'll be able to jump into questions here for just a minute. And the invitation I wanted to give you again is to tell us what you think of this feature. So you should be able to go to any of your past orders right now and upload your own file and do the visualization. So I'm really eager to hear how this works for you and what else you'd like. And if you need inspiration for what else you might see, I'm going to also point you to our Plasmid product. So the results page for that has quite a bit of visual information. The summary at the top has these nice quick maps of each plasmid, and then down below you can do the comparison, and these happen to be perfect, so not a lot of detail about the difference because there isn't any. But if you play around with your own data, you can see that there are some nice features for zooming and for getting more detail about any of the variants. And so, let us know which of those features you really like and you want us to add over into bacteria. All right, with that, I'm going to stop recording.